Welcome to all after the tea break. Happy to announce we have our own ex-HOD of Botany Department and Vice Principal of our college, Mrs. Manjari Barve ma'am. We welcome you, ma'am. We have begun with our second session by Dr. Samir Damare sir, Principal Scientist of NIO, Goa. On behalf of Department of Botany, we would like to welcome sir with a bouquet of flowers. I request Dr. Shilpa ma'am to kindly introduce our guest speaker.
Good morning. I welcome you, sir, for this national seminar. And I take this opportunity to introduce Sir. Dr. Samir Damle is Principal Scientist in Biological Oceanography Division of National Institute of Oceanography, Goa. His area of interest are molecular biology, genomics, proteomics, marine biotechnology, wherein he has guided 28 students till date. Dr. Samir completed PhD from NIO with work on interesting group of deep sea fungi under guidance of scientist Dr. Chandralata Raghukumar and joined NIO in 2009, and he is recipient of CSIR Young Scientist Award and Senior Scientist Award from Microbiologist Society Award in 2010 and 2020 respectively. Sir holds a patent in process of production of low temperature alkaline proteases from deep sea fungus. I request Sir to tell us more about them in today's session. Dr. Samir has 46 publications to his credit and published in reputed national and international journals. We welcome you, sir, with this short introduction, and I request Dr. Samir to take over the session.
For slides also. Yes. Good morning, everybody. Yes, morning you heard Belle Domza speak about how to identify fungal species. So now we will go further ahead. You know what fungal species are and how we identify. Now we'll look into the molecular aspects of proteomics. So I have started from basics into some difficult molecular biology things, and then I'll be talking about what we are doing in the lab with regards to proteomics and mycology. Okay, so I have a proteomics lab at present in NIO. If any one of you are interested, you can come anytime and have a look at it. Right from basic growing of the cultures to protein sequencing, identification, we do all that. So proteomics is not very difficult as people talk about, but then it is difficult to work with because of the degradation things what we see, because there are a lot of proteases in the cells. That's the reason people say proteomics is difficult. But if you take care of that, it's very easy to work upon. So both the aspects of genomics and proteomics started developing, but then genomics went way fast ahead. You will see that in my talk, why I say that way fast ahead, but proteomics is lagging behind. So these days, anything we need to confirm with Google. That's our whole and soul Maharaja. So if you see there, you will see a vast difference between the genomics and proteomics. You can confirm the numbers what I'm putting here. And then if you go further down, when you say about fungal genomics and fungal proteomics, it's really further down. There are, if you see, not more than 20 lakh hits come. That's because there are not many labs and many people working on that because of various reasons. So this is the level of biological information when you talk about. So this is your genomics or the DNA. Then you have mRNA, that is your transcriptomics. Then you have proteomics, then you have metabolomics, which ultimately gives the pathways, networks, the things which we don't like to study in the biochemistry. So all that are into here. And ultimately that leads to the populations and the ecology, which we are all part of. So this proteome term was first coined in 1994, and it was not some big scientist who coined the term. It was done by this Mark Wilkins as a part of his PhD thesis. So when he was doing some work, he gave this name as proteome, and that is how people started using it. So you don't need to be big scientists to put any new terminologies. You can do it in your BSc, MSc, PhD dissertations also. So basically, proteome is a protein complement encoded by a genome. So the genetic material what you have, it ultimately gives rise or gives to the proteome of the organism, which we study in the proteomics. These are some of the fundamental differences between the proteomics and genomics. This is where you will understand the complexity or the easy to learn DNA. When you talk about the genome, you have the DNA, which you are studying. The genomics analysis you do with mRNA, which is ultimately going to give the proteome, which is the coded mRNA, which ultimately gives rise to the proteins or the peptides or the amino acids, which are with the modification, without modification. Don't go into all those details. Genome is linear. It's like single stranded or double stranded. So you have lengthwise. When you talk about proteome, the main difference comes, it is having a 3D structure. So you have a secondary structure, you have a tertiary structure, you have a quaternary structure. The protein is mainly the folds which are defining, and the function of the protein is defined by the folds or the active sites which are there. So this is when you say about the genome, it's a encoded information which needs to be decoded. And proteome is partially encoded. So you have got some solutions with regards to and some understanding. What are the molecules and what are the functions it's going to carry about. If you go further little ahead, little more complex it becomes. Gene or RNA dye. Gene is very stable. Like when you dry a DNA and you can ship it at room temperature and again put water into it, you will get the same DNA out of it. Like when we send for sequencing, we dry it in vacuum, not in the oven or anything, vacuum dry, and the DNA remains intact. But that cannot be done with the protein, because the moment you are going to dry the protein, it is going to destroy the 3D or the quaternary structure of it. That is why protein will lose its functionality. So you cannot do that. That's the reason handling of the DNA is comparatively easier, cheaper. You don't need dry ice shipping or the liquid nitrogen shipping to send the samples out for sequencing or any other information. The DNA sequence is well established. You have something called as a pocket sequencer these days. It is called as MinION. It's like a small pen drive. You can put it in your pocket and the sequencing is going. If you connect to a simple laptop or even mobiles, high-end mobiles, you can do the sequencing. So the technology has become that cheaper and that easier. That cannot be said with regards to the proteomics. It still needs very complex setups and very expensive setups. For the genes, you have the DNA arrays, microarrays, what people talk about these days when you are doing any kind of screening worker. In terms of proteins, you have some protein chips or antibodies arrays, but again, they're really expensive and the life is very short of this. And the most important thing with regards to the DNA and protein differentiation in DNA, you have something called as a PCR amplification. So you have a small gene which you need to amplify and make copies of it. You can do it in vitro in the PCR machines, but you cannot do that for proteins even now. So you have to depend on the number of proteins which the cell is producing. So the proteins which are at low concentration in the cell, they're very difficult to study. So you need to have more number of cells. But the same thing doesn't apply for your DNA, because moment you have a known sequence and the primers are done, you can amplify n number of DNA copies and you can do the studies. So that is the main difference between the DNA genomic studies and proteomic studies, because of which genomic studies have gone way ahead as compared to the proteomics. But then proteomics is ultimately the ultimate function. It gives the living function. Whereas DNA, it could be from living or non-living organism. So even then it's difficult, but people are still doing both the things together. This is some of the major timelines with regards to the development of proteomics. So some of them I'll just highlight. See, DNA sequencing was established in 1977 using Sanger's method, what we study in our BScs. Then 1982, we had this something called as isoelectric focusing. In biochemistry, I don't know whether you have studied the isoelectric point of the proteins or the enzymes, which is called as pi value. Have you studied or no? Yes or no? Okay, good. Then, as the time progressed, there were different kinds of mass spectrometers which came into picture, and that is where the proteomics started increasing further. And now we have come at a stage where, as I said, the DNA sequencing has become very, very cheap, but then protein sequencing is still stuck in the mass spectrometry. You need a mass spectrometer. We have two mass spectrometers in NIO. If people are interested, there's one LCMS Q-top, there's a multi-top, which is being used for all these protein studies. I like to show this always because that's the difference between the DNA and the protein. Sorry. Yeah. So DNA, if you say that I have studied a DNA, if you take it from an egg or anywhere else, your DNA is going to be same. It's not going to change. And you can say that 100% DNA sequencing has been achieved. That doesn't happen when you say about a protein. Proteins, all the proteins of in a cell are never expressed at any given time. Whatever is required, because nature is that way very energy conservative. Whatever is required, only that is expressed or that is produced in functional forms. So when you say about I am studying the proteomics, you have to mention at what stage of the life cycle you are doing it. Because if you see this is a butterfly life cycle, you have an egg, you have pupa, you have a larva, and then you have butterfly. So a person who is going to see only physical traits, larva and caterpillar, butterfly are very, very different. There is no comparison. If you tell this to small kids and say, no, this is same, they will not trust you. So that same thing happens when you're talking about the DNA and protein. The DNA sequence of both will be same. There won't be any difference. Only thing, some genes will be expressed, some may not be expressed. And those genes expressed are going to make a difference in the proteome or the protein profile of that particular life stage. So it's basically the same genome. It's the expression of the proteins at any given time, which is differentiating a caterpillar or a pupa into a butterfly. So that's why we say that proteomics is basically an attempt to describe the biological state and the qualitative and quantitative changes which are occurring in the life form. And these extracellular biological materials and different conditions are further give an understanding into the biological process or the biological stage which the culture or the organism is going through. So this is the general strategy which we follow to study the proteomic analysis. Afternoon, we'll be having a small demo about the STS stage. So I will be doing a demo. This part onwards, what you say, the secondary. So here, this is what we use normally. You have like, when we talk about, say, I'll talk in terms of microbiology because I'm basically a microbiologist. So we grow the cultures in the lab. You need to break open the cells. If you are going to study the cell wall or the membrane or the cytoplasmic proteins, you need to get the proteins out. So for that, you need to break open the cells. When the cells are broken open, the most important thing is they should be in soluble form. anything in insoluble form you cannot analyze. Then they should not be degraded. Because what happens inside the cell, you have your proteins and you have proteases also, because ultimately the life cycle, the catabolic and anabolic processes are going on, which degrade, but they will not do it in the cytoplasm. Moment they come outside of the cell, they will attack and they will try to degrade the protein. So you need to take care of the proteases. After all this is done, then you can start analyzing of the protein, which is done. This is the most common used technique in 1983-84. It was started, called as 2D PAGE, two-dimensional polyacrylamide gel electrophoresis. So what we mean by 2D is that you have first dimension separation based on your isoelectric focusing, and the second dimension separation is based on the molecular weight. So because of that, you get proper separation, because what happens, many of the proteins, they have similar chemical structures or the charges, because of which you have something called as isoelectric forms. So you could have same size, but the charges will be different. So when you use both the strategies to separate the proteins, you will get better separation. And some of the good labs who do 2D gels, they have managed to identify and separate up to 10,000 proteins on one protein gel of around 15 centimeters by 15 centimeters. So that powerful technique this came into existence much long before we could think of. And then once the protein is separated, then you can do all this digestion of the proteins and then the MS fragmentation and then identify. That is called as peptide mapping. I will not go in detail of this. So this is how it looks. Your 2D gel. So each spot here is representing a protein, which is separated based on, again, as I said, on the PI, that is isoelectric point, and the molecular weight of the protein. And then the visualization is done using different kind of stains. So it could be a Kumasi, or it could be a radio stain, or it could be a immunofluorescent stain, or it could be silver staining. So for that, many of the methods are available. As I was saying initially, that proteins are 3D structure, and the bonds which are there, they are not so easy to break. So you need to have special harsh treatments which will not harm the protein structure as such, but it's going to break open the bonds. So these bonds are basically disulfide bonds, SS bonds, which are very strong and need to be broken. So most of the time, urea is used for making these bonds and to keep the protein in the soluble form. Because the moment the protein is going to become insoluble, it's going to precipitate out. And when you do the further downstream processing, you'll be losing the protein sample to the separation procedure only. So these are some of the, this is an example of the sample buffers which is universally used. You have some urea, thiourea. So these are used to keep the protein in the soluble form. Then you have some surfactants, which will again help to keep it in. And then you have some reducing agents, which will not allow the rebinding of the disulfide linkages. After your separations are done, these are different detection technologies which are used for the proteome analysis. So this could be organic dye or silver-based methods called as Kumasi blue or silver staining. This is most commonly used in labs. Then you have radioactive labeling. You need special labs for handling this. Reverse stain methods or fluorescent methods. Fluorescent methods, you can detect proteins up to 1 picogram also. So it's extremely sensitive and you don't need any special labs like radioactive labeling. Radioactive ultimately is the final thing for detection, but then you need special labs for it. You can't use it anywhere. So that's the general strategy what we use. I will now talk something what we have done. One of a PhD student has done during our PhD. Proteomics as a tool to understand the effect of elevated hydrostatic pressure. That's the topic when my work was being introduced. I have done my PhD in deep sea fungi. So I started looking at whether fungi are present in deep sea or no. That is where we started. And now we have reached a stage where we know fungi are present in deep sea. So we started going, next question is, if they are present, how do they survive in all these extreme conditions? So this is my work area. What you see marked in pink, that's India. So it's beyond equator, somewhere down in 13 degrees south. I was lucky to be part of a project called polymetallic nodule mining, deep sea mining, which is funded by Government of India, under which we did a lot of expeditions in this area. So the average depth from where we have got these samples are around 5,000 meter plus. And in spite of that depth, we could get growing fungi from these places. This is thanks to this. This we have in our laboratory. It is called as a pressure simulator, which is used to cultivate the pressure in the native conditions of the deep sea. When I say native conditions, with regards to pressure, what happens is as the depth increases, as you go down, for every 10 meters of your depth increase, you have 1 atmosphere, 1 bar pressure increase. The room where we are sitting now is at 1 bar pressure. So that's what we call as atmospheric pressure. As you go up to the mountains, the pressure decreases. And similarly, when you go down to the sea, you have pressure increases. That happens basically because of the weight of the water. Like every 10 meters, the weight is there. And then as you go deeper down, Mariana Trench is the one which is the deepest, 12,000 meters. So you have 1,200 times more pressure than what we are sitting in here now. But in spite of this harsh conditions and the pressure, organisms are able to survive. And we have got some fungal cultures in the laboratory which are capable of growing at around 400 bars also. So all those growth we carry out in this cylinder here. Yeah, if you see the names of this, what we got from the deep sea, these are all simple names, or you see Aspergillus penicillium and all. Yeah, we did studies with regards to that, and we have proposed theories like how the terrestrial forms are adapting. I will not go in details of that here. So as I was saying you, we established the presence of fungi in the deep sea. We could see that they are able to grow under that pressure and the temperature. When we looked into the aspects of what they are doing, we proposed some theories with regards to the aggregation that is out there, doing the carbon sequestration and some enzymes which are being produced. But then there was one question unanswered which we were not aware. That's how we started that work. Like, how do these fungi or these organisms capable of surviving in these conditions? Like, what is the difference at the molecular level? Because these are the same Aspergillus, Terius. Moment you say Terius, it's a terrestrial. But in spite of that, the Aspergillus, Terius, what we got in mangroves was not able to survive in this deep sea. But the one which we got from deep sea was able to grow. So what is the difference between the two? Can we train this mangrove fungi to grow in that pressure? That is called pressure adaptation. So all that to answer, we started looking into the adaptations which could be there. So when you talk about adaptations, it could be in the form of the morphological adaptations or the physiological adaptations. In the morphological adaptation, these are some of the adaptations we could see in the fungi what we isolated from deep sea. This is your center classical Aspergillus, that's what picture we see in the textbooks. But these are the different forms what we could see directly under microscope from the culture which were growing from the deep sea. But then the most important thing what we realized was, apart from this morphological, ultimately it is something is happening at the molecular level or at the proteome level, and expression of proteins, and that too the stress proteins could be one of the reasons, and the different kinds of stress proteins these cultures are producing and allowing them to survive in these deep sea conditions. Okay, this was a workflow what we followed for this work. We grew those all cultures in different hydrostatic pressures at that pressure cultivation vessel what I showed. We again broke open the fungal cells. They are very difficult to break because of the chitin in the cell wall. Bacteria you can just boil in the trace and the cells break open. That doesn't happen in case of fungi. You need to have special breaking mechanisms for that. Ultrasonication sometimes works, it doesn't work. Most of the time people used to use this sand which is there on the beaches. Acid wash sand, and you grind that acid wash sand with the fungal biomass in liquid nitrogen. That is how we could break open the cells. We use something more harsher called as a homogenizer with zirconia beads. And zirconia beads are neutral charge, because if you use glass, again you are going to get attraction of the DNA and protein to the glass and you'll be using it. So we use neutral charge zirconia beads in our lab to break open the cells and get the cytoplasmic and the cell wall proteins out. And this is the platform which we use for carrying out all the machine. This is called as a LC-MS QTOF. LC stands for liquid chromatography. That's in HPLC what we study. MSMS stands for mass spectrometry, and TOF stands for time of flight, and Q is for quadrupole. So it's basically assembly of 4 different machines which are put together and called as LCMS QTOF. So this is the one which we have used extensively for all kind of proteomics which we are doing in our laboratory. We are having this setup now from 2012 onwards. Almost 12 years we'll be completing with that. And a lot of people have used it successfully. I will not go in the data details of this. Yeah, this is what you see basically. This is the output of the machine. What you see, some lines and all you get, doesn't make any sense unless you put it in the software, which is ultimately going to match the spectra to the known spectra which are submitted in the databases. Like we are using databases like NCBI most of the time, or there are other specific databases also like SwissProt and all, which are catering to only some proteins which are there. But NCBI is one of the most used databases which will give you, and ultimately this will give a match to some protein which is already described. So the limitation at present what we have is we are dependent on the database to identify. Unidentified ones is not easy to get. Then you need to go something called as a de novo proteomics. Very complex to cover in this lecture here. So this is what we get as an output of the statistical analysis with regards to the expression levels. So when we grow the culture at one particular pressure and the same cultures grow at different pressure, what we do is we try to compare the proteins which are produced in one pressure to another, and then we talk in terms of the log4 changes in the protein expression. And there are some proteins which are produced only exclusively when the pressure is given. So these could be the proteins which are responsible for the survival of these cultures. And then further we have gone ahead and identified the specific proteins and seen that these are nothing but the stress proteins. So initially when stress proteins were described in 1962 somewhere, they were described from this fly, Drosophila, and they were called as heat shock proteins because they were described basically against heat shock. That's why they were called as heat shock. But as the time went on, the terminology changed and it went from heat shock proteins to stress proteins. So any kind of stress, like now you are sitting here and suddenly electricity goes off, you will be having a heat stress there. So there also your body will start giving. That's the reason these proteins are at that particular moment. So your sampling is very, very essential when you want to catch this protein expressed or the genes expressed. So these changes are monitored, and based on that we can pinpoint that this particular protein and this particular pathway is getting affected by the pressure or the temperature or the stress you are talking about. Here we studied pressure and temperature, two stresses, because as I said about the pressure also, the temperature also changes. If you go to the sea now and check, the surface sea surface temperature will be somewhere at 26, 27 now in winters. And if you start going down deeper, beyond 1,500 meters, it goes at 4 degrees. It's like a huge refrigerator down. So it's completely, but in spite of that, activities are going on. So that could be classified as cold shock proteins because it's against the cold. So any kind of shock, we say as stress proteins, and these are the ones which are responsible. So the function of the stress proteins, it could be protection of this the enzyme structure or the protein structure, the 3D structure. So other words, they are also called as chaperones. I don't know if this terminology is yet covered. So the stress proteins or chaperones are the ones which are responsible for that. Like that, we identified around 12 chaperones which are exclusively expressed under the pressure. And further, we need to, if you are studying, then you need to do the gene cloning of that. And that was not done. We stopped at identification of proteins only in this work. Yeah, when I say further is that you need to do all this kind of work. We did genome, we did proteome, then we need to go back and see that which is the gene which is responsible. There are some experiments which are carried out, not in our lab, but other places they have done with regards to Saccharomyces cerevisiae, commonly used yeast, where they have taken the genes which are responsible for this pressure temp tolerance or temperature tolerant. They have managed to clone it, get it out, and put in another cell of different yeast, and they could see that there was a difference in the growth with regards to the stress tolerance levels. So if these kind of things are there, say the fermentation industries and all, they're going to help. So somewhere or other, you need to have the basic information available before we do the application. So these are the different application-oriented omics which people talk about, and these students are very much interested in this. So you have genomics, transcriptomics, proteomics, metabolomics. We do all this in our lab, the first three. Metabolomics we don't do yet. It's going away little from proteomics, but these are some of the important scientific technological advances in the marine microbial ecology, which we all, I don't know, microbiology you must have heard of plate count anomaly, right? BSc. Yes or no? Okay, all of you are exclusive, pure botany people. Some micro are there. So you know what is plate count anomaly, right? So microbiology started somewhere from plate count anomaly, and which we have reached a level of sequencing and able to mutate a single base pair. So it's gone to that detail. So these are the different key points which are available about the DNA sequencing, Sanger sequencing started somewhere in 80s. Then you had 454. You have now everybody is talking about Illumina sequencing. And as I was saying, sampling is one of the most important thing when you are doing any kind of this biological or environmental studies. So in the cell also, and you go in the oceans also, when you are trying to do the sample collections, it has to be exactly the point which is required and what we needed. So these are some of the sampling expeditions which were covered, and this was the first global attempt to study the genetic diversity of the oceans with regards to the bacteria, fungi, viruses, plankton. So most of the things are covered under this. I hope you all know Craig Venter. So he's the one who competed with US government and did the human genome sequencing. So when he finished, I don't know, this is an anecdote which people talk about. When he finished the DNA sequencing of human, he had a lot of sequences with him, around more than 600 plus, which he didn't know what to do. So people said that he ventured into the oceans, oceanography. That is how we started this called as Global Ocean Sampling Projects, which ran from 2003 to 2010, wherein he covered all these areas, and a lot of sampling was carried out by his team and by scientists who were interested. So he did not charge anybody anything. Who were interested could go and collect the samples in these expeditions. And that has contributed hugely to the bacterial and the fungal and other plankton sequences in the NCPI as well as the other expedition projects which were there. This is the second expedition which was carried out as to Tara Ocean. This also looked into all the microbial components, the bacteria, viruses, and the fungal and the plankton, wherein more than 40 million genes contributing to all these organisms have been deposited, and it's open, available to everybody to look into for data mining. Under this, this is called as a KM genes database, where this kind of genes are deposited, and if you are interested, you can download the genes and you can look into it. No restriction on that. Anybody can access this data. So with the proteomics in the lab, now we have moved on to the metaproteomics. So at present, we are doing the Indian Ocean Proteomic project in the laboratory. These are some of the examples which are available with regards to the ocean metaproteomics, but not many are available from the Indian Ocean. So that's the void which we are trying to fill in. And when I talk about the metaproteomics, when I was talking about the fungal proteomics, again, ocean metaproteomics, it further goes down. You really don't do any work into it, because it could, when you talk about meta, is basically from the uncultured part. When you talk about the fungal proteomics, you know that the proteins are coming out from fungi. So you could identify them more easily. In metaproteomics, it doesn't happen. It's a mix of all the proteins. It could be from the bacteria which are present in the seawater. It could be from the fungi. It could be from the yeast. It could be from the plankton which are present in the seawater. But then it is very important to look into these aspects now, because ultimately that is the one which is giving you the signal of the live processes which are going on there. It could be in the term of the biogeochemical cycling or the microbial responses when we talk about the ocean warming and all the climatic changes. So ultimately these indicator organisms which are present in the form of the microbes in the seawater are going to get affected, and they will be giving us signals to understand. But for that, you need to have a baseline. You need to know what was there initially before it changed. So that's what we are trying to do in this project. But then when we talk about the vast oceans which are there, the challenges which we face in terms of this technologies are basically environmental sample acquisition and extraction. When you say about any lab-based work, you can grow the culture in the laboratory, you can get as much as you want, and then you can do the studies. That doesn't happen when I was talking about the comparison between PCR and proteomics. The same thing happens here also. In the lab, you are in control of your experiment. You can grow whatever cultures you want and get. But then in terms of the naturally occurring organisms which are there, like if you say in terms of the fungal component, you will find a lot of unicellular yeast in the seawater. But the same cannot be said about the filamentous fungi because they need some substrate to grow into. So that's why you need to filter more sample, or you need to study. Like when you talk about the Mycology, they will be more in the sediment, not in the water column. So similarly, you need to collect the sediment from there. So different samplers which you use for this kind of work are. When you talk about the water sampling, this is what we use. This is called as a Niskin sampler, Niskin bottles, which are attached to a CTD rosette. So these bottles, what you see, are normally open at both ends. They are sent down at whatever depth you need to collect the water. There's a trigger on the depth. When you press the trigger, the water sample gets trapped in this bottle, and this both ends close, and that is how you get water from that particular depth, and that is got back to the lab and work. But then, as I was saying, if the number of the naturally occurring organisms is less, this sample will not be sufficient. So you need to have special samplers. So this is called as a Gerald barrel, which can collect up to 250 liters of water from one particular depth. So you don't need to keep on putting that up and down because it's extremely expensive. Every minute spent there is counted upon. When we go on any oceanographic cruises, the cost which we incur is like 17 to 20 lakhs per day for any expedition which is going. So it's expensive sampling. You need to have minute by minute and liter by liter account of that. This is something new which we have been using at present. So instead of you getting the sample and doing the filtrations on deck, this is a pump here which you can lower at one particular depth. It will do the filtration there only, and then you just get the paper out. This is called as a Macklin water transfer system. This can, depending on the battery life, you can leave it down for 3 to 4 hours, and then you get that much of sample. Like 3 to 4 hours, it will easily filter 300 to 400 liters of water sample, and it will be in situ there. Like I was talking about the pressure, like what happens when we get the sample up. Most of the time, the unicellular forms, they survive, but that cannot be said about the multicellular forms. Like say starfishes or even fungal filaments we have seen, they break open and the contents are coming out. So to study these kind of organisms, you need to study them in native condition. So this is the advantage of this kind of filtration units, where you can do filtrations at native condition and get the filter paper out and then do the studies with that. So this is, we have started using, but then again, this is time consuming because only one person can get the sample at a time. And if you are more interested into the benthic organisms which need support, like nematodes are there or filamentous fungi are there, you need to look into the sediments. That's the soil which is at the sea bottom, below the water column. So these are the samples which we use for that. It's called as a spade corer or a box corer. This is the box which gets the sample inside. This is how it looks, filled with the sediment sample. And then you get this kind of cores. Now this area where we sample normally, this is 50 centimeters. So each 1 centimeter, it takes around 1,000 years for it to accumulate in that area. So when you talk about this much 30 centimeters, this is 30,000 years old sediment, which are getting and working, studying in the lab. And the deepest from where we have recorded fungi in our lab was 0.42 million years old. We got it in Chagos Trench in Indian Ocean in 2001 expedition. where we managed to grow that fungus in the lab. Ultimately, it was Aspergillus only. But then it took very long. It took more than 9 to 10 months for it to start growing in the lab. It had to be really trained to grow in the laboratory conditions. It was Aspergillus sidovi, which we got. These are different kinds of samples to collect the sediments again. But then after getting these samples out, getting the proteins out from this is very difficult because there are a lot of non-proteinaceous matter. It could be in the form of polysaccharides, basically, which create a lot of hindrances in the extraction of the proteins. And that is where you need to develop the extraction strategies such a way that you get your protein out and it remains intact also. That's where we play with the composition of the buffers, the urea, thiourea. Those are the basic components, but you need to increase or decrease depending on the complexity and the presence of the humic material. You don't have that much of humic material in the deep sea as you get in the garden soil, but then yes, it is still there, and that causes a lot of problems in the protein extractions. So biological composition of these samples can be largely unknown right in the extraction because ultimately you're getting unknown. As I said, in the lab when you grow, you know what the fungus is going to give, but that cannot be said in terms of the sediments, where we'll be having a mixture of organisms, which ultimately all those proteins, once the cells break open, they're going to interact with each other, and then you need to separate them out and then study. So these are some of the challenges which are present for both the separation as well as the mass spectrometric components, the complexity of the seawater samples. So this, once the proteins are separated and data is available, it will give us a metabolic record of the state of the key microbial components within that particular spot or that geographical time frame when we have collected the sample. And this will help us looking into the how amino acid data interference with the proteinic data is occurring and how the proteins have evolved. Because when I talk about the samples which are way below down at 30,000 or 50,000, we don't know whether the same proteins are there, same structures are there, whether they have evolved with the time. So all this could be understandable. That doesn't happen in terms of DNA, because DNA is going to remain almost same, but then the structures of these proteins with regards to the functions might be the one which is changing with the pressure and the temperatures which are there. So this is the present state of the proteome map of the oceans. If you see Indian Ocean, as of now, there is no records available. This is what we are. This is one portal which is being maintained by Oxford Institute of Oceanography with regards to all the proteinic samples which they have done so far. But then no records are available at the Indian Ocean. And to fill that gap, we got a project in 2000. We started a project with regards to the genome and proteome mapping of Indian Ocean. So that protein data, whatever I presented now, is a part of all this big project which we did. And we carried out a huge expedition in 2021 of 89 days sailing, where we collected more than 700 samples for doing the genomics, proteomics, and transcriptome studies of the water samples, sediment samples, the cultures, bacteria, fungi, plankton. And we are trying to link it together to all the baseline data which could be recorded. So this is the sample from the close-up what you saw in the previous slide. This is called as a Lispin or rosette sample. This is called as a multi-plankton net, which is used to collect the planktonic life forms, zooplankton, phytoplankton from the water column. And these are the sediment samples. Yeah. So around 56 to 60 centimeters we can collect in this particular tube. And again, the same protocol we follow to generate the proteomic data, whether it could be cultured or uncultured. Ultimately, it is basically the protein or peptide separation, which we carry out on this machine, on the LC. And then we identify the result proteins in the term of the spectra, match to the database. Now, what we have started is we are studying our own databases because there is no information available from that particular area. And we are collaborating with this metaproteome platform called as Galaxy T for doing all this proteome analysis. These are some of the outputs. I will not go in details to explain what is this, but This is the kind of output we get with regards to the peptide-centric taxonomy. So based on the proteins also you can identify, you can go back and say that this particular protein has come from this organism. It's not always other way, like you know the organism and then you identify the protein. So you can always have the reverse engineering where you identify the protein and then you can match it to the organism. So that is called as a peptide-centric taxonomy, where you identify the proteins and then assign them to a organism. Then within this, we are doing some comparison with respect to, like if you see this area, this is one of the most productive and we have different geological settings here. This is called as a 90-degree ridge. Geology students would know it better. It's a mountain basically at 90-degree east, which is like suddenly come out from 5,000 meters to 1,500 meters depth. So we are doing a comparison along that to see what are the changes which are occurring along that. So this is the data into that, which is giving again the peptide-centric taxonomy, where we could see the change in the organisms right from north to south. So we are hoping that by another 1 year or so, we'll be able to fill this blank which is seen here. Maybe 2-3 years down the line, when I come for another talk, I'll be able to present more data with regards to this and all the metaproteomic work which we are carrying out in the laboratory. So if any of you are interested, you can just Google out this stress biome. There is a documentary which is done by Doordarshan about our expedition. 2 hours English and 1 hour Hindi. So these are the links for that. You will have an idea what exactly we do on the ship, right from the sampling to the DNA extraction, to the protein extraction, to the RNA extraction. We do all the culturing work and all the molecular extraction works on board itself. We don't get the samples back to the labs. We have well-established labs in the ship itself where we do all this analysis. Next expedition, I'm trying to carry that Minion sequencer also, so you can do the sequencing also on board. So this was my students who stayed with me on the expedition, and this was the whole team. So we were together during the COVID second wave, which was going on in Goa from March 2021 to June 2021. So three months we were safe in quarantine on a ship, and we came back when the COVID wave got over. So these are all my students and my PhD supervisor. Yeah, one of them is sitting here, Amara. Okay, yeah. Thank you.
Send the link on WhatsApp to you, and I can post it afterwards.
For the online participants, if you have any questions, you can ask or put it in the chat box. I'll try to answer them. My email ID is samir@nih.org. If anyone is interested, they can write to me also later on. Especially for the deep sea cultures, as I showed you a picture, we have a pressure simulator. So we grow it at the same pressure from which they were isolated. Like say 5,000 meters, you have approximately 500 bar of pressure. So we put the cultures in that pressure. Then again, another modification which we do in our lab is that if you try to grow them in the conventional media, like your malt extract and all, many times you have seen they don't grow because they are coming from a condition which are quite oligotrophic. You have very low nutrients there. And if you try to give them and grow them in the rich media, sometimes it comes as a nutrient shock also to them. So we dilute the media. Like sometimes we have gone to dilution of 1:100 also. Then another thing which we tried during the thing is we got the sediments from that particular area. We prepared a sediment extract. So the nutrients, whatever they are available in that nutrient, we filter sterilize them and we made a media of that. And then we found that the growth was better in that as compared to even lab media. Then we started using those only for all the experiments. So sometimes the native sediments support what we call as common soil agar, if you do. They support the growth better than the microbiology media what we use in the lab. Yes, you can refer my 2008 paper, Microbial Ecology, where in detail on my thesis, which is available online in NIO, where we have given the details how the sediment extract is prepared. Yeah, there is one question from Aditya saying, can we identify novel antimicrobial peptides and enzymes with biotechnical potential from fungal proteomes? Yes, we can do that as long as you have the complete proteome which is described and it is available in the database. We are doing some gene mining with this regards for some of the deep sea cultures in some different project, which was from the biotechnological aspect, where we are working basically with the small peptides. We have not yet looked into antimicrobial peptides as such, but as of now, we are doing some identification of small peptides which could be used for that work. But yes, it can be done. Meenam Patil has raised the hand. Please, yeah, ask your question. Yeah, there is another question which is talking about the fungal proteome change during interactions with other organisms such as bacteria. Yeah, that would be really interesting subject to look into, but we have so far not gone into the interactions because it is as of now very difficult to get biomass for the pure cultures only. And doing this experiments in the pressure is not that easy because you don't know how the organisms are going to behave, especially the plants. Because plants, when you talk about an deep sea, you don't have any active chlorophyll activities going on there because deep sea is always in dark condition. So you don't have any plants there, but then we have done some interaction studies with regards to nematodes and deep sea fungi, where we have seen that these nematodes are basically eating this fungi and some of the pathogenic fungi are infecting the nematodes. So some experiments are done by one of the PhD students in other lab with the same samples what we got from this area, around 5,600 meters. But the main difficulty in this is to get those big microorganisms difficult because with respect to fungi and bacteria, we can always grow them in the lab once you have your right conditions. Nematode culturing and other microorganism culturing is not yet possible under pressure conditions. Yeah, we normally do the dating with regards to the paleontology. We do datings from forams because these foram records are available for this area, and based on that, carbon dating and all mostly doesn't work for this step because that's only up to 5,000 years old. But for this, we use foraminifera dating techniques with the help of the paleontologists which are available who are working in this area in our labs. Any question from the students? So I can ask.
So this is why
It is on YouTube, DD News YouTube channel. I can share the link with you. Patterns of deep sea fungi differ. There is a difference. The question is about how do the protein expression patterns of deep sea fungi differ from the shallow water counterparts. We have carried out these experiments with the Aspergillus terreus, which we got from the deep sea, and then Aspergillus terreus, which we isolated from the mangroves. And we could see a lot of differential expressions in the proteins. Just referring to my student's thesis 2018, Akhila Krishna Swamis, he has done a lot of examples in this because more than 300 to 400 proteins we could see the differences. So for the details, please refer to that. Yeah, there's a question with regards to NI offering postdoctoral research for PhD holders. Yes, we do, but we don't have any scheme as such for postdoctoral research. You should get your postdoctoral fellowship from the funding agencies like DBT or DST. DST Inspire, we have a lot of faculties at present working. After PhD, you get that. So you need to contact the scientist and then get the approval and apply to the funding agencies. Then we take. Otherwise, you need to look into the advertisements which are coming under the project. But then postdoctoral fellows we rarely advertise. It's basically the people who are coming with old fellowships. They can come for postdoctoral research.
I request Manjari ma'am to present a memento to Dr.